Neanderthals are an extinct type of human that lived in Europe and parts of Asia between 200,000 and 30,000 years ago. They were stronger and stockier than us, and their heavy-browed skulls could accommodate a large brain. They are the hominin form most closely related to modern humans. Based on DNA sequencing results, it appears that Neanderthals and modern humans had a common ancestor who lived about 660,000 years ago. Sequencing the entire Neanderthal genome has been made possible by new technologies called next-generation sequencing. Neanderthal DNA was extracted from bones discovered in a cave in Croatia. Ancient DNA presents a number of sequencing challenges. The primary problem is that over time DNA degrades. 100,000 years is considered too old to contain useful DNA. But these bones were 38,000 years old and fairly well preserved. Investigators could extract Neanderthal DNA fragments that were 50 to 100 base pairs long. Another problem with ancient DNA is that the nucleotides themselves may be chemically modified. Over time, a cytosine can become deaminated, converting the base to uracil. In sequencing reactions, uracil is read as thymine, not as cytosine. In order to achieve acceptable sequencing results, it would be necessary to sequence each part of the Neanderthal genome at least 5 times to exclude all such errors. Microbial contamination presents yet another hurdle. Most of the DNA found in Neanderthal bones is from microbes that colonize the body during decomposition. To enrich for Neanderthal DNA, some of the microbial DNA needs to be removed during the sequencing technique. In this case, investigators used restriction enzymes that digest only microbial DNA sequences, a method that is not frequently used. Lastly, contamination with modern human DNA is a significant problem. Modern human DNA can dominate the sequencing reactions and could potentially exclude the ancient DNA altogether. As part of a strategy to avoid contamination, laboratory reagents and instruments with traces of DNA from modern humans is avoided. There are several types of next-generation sequencing methods, and each works in a similar way, making use of the polymerase chain reaction to amplify the DNA to be sequenced. The method used for sequencing the Neanderthal genome is called pyrosequencing, an early second-generation method that is no longer widely used. If the DNA starts out intact and long, it must first be broken into pieces that are less than 500 base pairs in length. Short oligonucleotides, called adapters, are ligated to each end. The DNA is then separated into single strands. The adapters enable the fragments to be attached to metallic beads. One of the adapters has a small molecule called biotin attached to its 5' end. Biotin has a strong affinity for a protein called streptavidin, with which the beads are coated. The ratio of DNA fragments to beads is set so that on average each bead has a single fragment of DNA. To get enough DNA to sequence, the DNA is amplified by PCR using primers that anneal to the adapters. This means that the same primers are used to amplify all the DNA fragments. The beads and reaction components for PCR are placed in tiny water droplets in an oil emulsion. On average, each droplet contains a single bead. Using these beads and the tiny droplets, many thousands of PCRs can be performed in parallel. Each bead becomes coated with many copies of the same DNA sequence. The beads, with their attached PCR products, are now transferred to sequencing plates with millions of tiny wells, each big enough to hold just one bead. The wells are also packed with sequencing enzymes. The method used for sequencing the Neanderthal genome is called pyrosequencing. All of the wells, each with their unique DNA sequences, can be read at the same time. Let's look at a strand of DNA from one of the wells. A sequencing reaction employs DNA polymerase, which uses a primer as a starting point, and the DNA strand to be sequenced as a template. Nucleotides with a single type of base are flowed over the plate one at a time, starting here with DTTPs. If the template DNA has an adenine-bearing nucleotide in the first position, DNA polymerase adds a DTTP to the growing strand and releases a pyrophosphate as the byproduct. Two enzymes, called sulfurylase and luciferase, convert the pyrophosphate into a flash of light, which is recorded. Pyrosequencing is based on detecting pyrophosphate. Unincorporated nucleotides are degraded by another enzyme, nucleotidase, and then the next set of nucleotides is flowed across the plate. Again, the successful incorporation of a nucleotide is accompanied by a flash of light. If no nucleotide can be incorporated, then no light is emitted from the well. The incorporation of two nucleotides results in twice the number of pyrophosphates and twice the amount of light. As the new strand is being made, the order in which nucleotides are incorporated is detected, so the sequence can be read as the reaction proceeds. The sequences determined for the Neanderthal DNA fragments can be aligned with those known from modern human DNA and overlapped with other Neanderthal fragments to produce a full sequence. By 2010, a draft of the Neanderthal genome sequence was completed, with some regions covered multiple times. The Neanderthal sequence is, like our own, 3.2 billion base pairs in length. Overall, Neanderthals and humans have genome sequences that are about 99.8% the same. Compare that to any two humans, who are 99.9% identical, or to humans and chimps, which are 98.3% identical. To learn more about the relationship between Neanderthals and humans, the Neanderthal genome was compared to the genomes of 5 present-day humans from all over the world. One San individual from southern Africa, one Yoruba from West Africa, one Papua New Guinean, one Han Chinese, and one French from Western Europe. In a model of human origins called Out of Africa, humans evolved in Africa, and then a subgroup of humans migrated out of Africa and colonized the rest of the world. Modern humans arrived in Europe about 45,000 years ago, and Neanderthals did not become extinct until about 30,000 years ago, so they must have lived in the same regions for several thousand years. Did they interbreed? To find out, investigators identified regions in contemporary human DNA in which non-Africans were different in sequence from Africans. For 10 out of the 12 of these regions, Neanderthals had the same sequences as the non-Africans. If there had been no interbreeding, non-African sequences would be no more similar to Neanderthal sequences than would be the African sequences. In fact, non-Africans were more similar to Neanderthals than could be expected by random chance. The conclusion is that interbreeding likely occurred, such that non-Africans inherited 1 to 4% of their genomes from Neanderthals.